Hello, testing. The Hong Kong Polytechnic University, PolyU, leverages its world-class academic and research excellence to develop innovative solutions for a more sustainable tomorrow. In July 2022, the PolyU Academy for Interdisciplinary Research PAIR was established as the university's central research platform, dedicated to providing world-leading, interdisciplinary solutions to address significant societal challenges. PAIR is composed of multiple constituent research institutes and research centers. Each research unit focuses on a particular research area and is joined by researchers from various PolyU faculties, schools, and departments. PAIR research areas include advanced technologies in manufacturing, good health and well-being, and smart and sustainable cities. PAIR researchers develop new materials, systems, technologies, and techniques that support industrial modernization, upgrading, and transformation, look into the factors and mechanisms influencing human health and wellness, and develop evidence-based solutions, and generate smart technology-based solutions for making urban operations and services more efficient, connected, and eco-friendly. We particularly value impactful collaborative activities in research and scholarship. PAIR researchers pursue research collaboration at different levels. In addition to interdisciplinary collaborations within and between research units, PAIR and its units establish partnerships with industries, governmental bodies, universities, and other external stakeholders. With internal and external collaborations, we have successfully translated PAIR-led innovations into commercializable solutions and practical applications, and established joint laboratories and research entities with external organizations, driving scientific breakthroughs, innovation, and technology development, and socioeconomic prosperity. PAIR's research excellence and research impact have been widely recognized at local, regional, and international levels. Our research outcomes have been published in leading journals, including Nature and Science. Innovations led by PAIR researchers have won prizes at various competitions and events, including the International Exhibition of Inventions Geneva. Many PAIR researchers have been named among the world's top 2% most cited scientists in their own areas of specialty. Individual researchers have been selected to the National Academies of Science or Engineering and as fellows of professional associations, and have been bestowed with local, national, and international awards and honors. Harnessing the strengths of PolyU's research and world-class scholars, PAIR will continue our unwavering efforts in research and scholarship, knowledge transfer, and collaboration to develop impactful solutions that benefit Hong Kong, the nation, and the world.
Okay, ladies and gentlemen, so good afternoon. Okay, welcome to the PAIR seminar. I'm Ka-Heng Wong, the Director of Research Institute for Future Food under the PolyU Academy for the Interdisciplinary Research PAIR. Okay, so today is my pleasure to introduce our seminar speaker, Professor Hélder SANTOS, the Professor and Head of Departments of the Biomaterials and Biomedical Technology at the University of Groningen in the Netherlands. Professor Sandro is a physical chemist with expertise in nanomedicines, biomaterials, pharmaceutical nanotechnologies, and controlled drug delivery. His work reflects how medicines are designed, delivered, and translate into real-world clinical impact. Professor Santos' research focused particularly on developing innovative nanomaterials for cancer, cardiovascular, and autoimmune diseases. He develops advanced nanomaterials such as nanopores, silica, and liposomes, which serve as smart platform for drug delivery, diagnostics, regenerative medicine, and therapies. Ladies and gentlemen, allow me to highlight two examples of Professor Santos' impact research. Okay. So first, in cancer therapy, treatments such as RNA medicines and immunotherapy often struggle to reach the right target in the body. So Professor Santos developed nano-based delivery system that protect these medicines, deliver them more precisely, and improve their effectiveness. So his work also explore combinations therapies, okay, where nanoparticle deliver multi-agents simultaneously to achieve better treatment outcomes. So Professor Santos has a lot of achievements. So if I want to spend a lot of time to go through, I think I'll need to have 1 more hours. So to cut it in short, okay, so without further ado, ladies and gentlemen, please join me to giving a warm welcome to today's seminar speaker, Professor Hélder Santos. Thank you.
So good afternoon, everyone, and thank you so much, Professor Wong, for the very kind introduction. I would like to, it's my great pleasure to be here in Hong Kong, my first time. And I would like to start to thank Professor Wong, Professor Chow, and Professor Kevin for hosting me here this week. It's a great honor and a great pleasure to speak to all of you here present, and also for the audience online. What I'm going to talk today is about what we do in our research. And the topic that I chose today to show you here is about cardiovascular disease. This is only one of the that we do. But before that, let me start to tell what my lecture is going to be about. So I'm going to first focus. Thank you. Give you an overview about our research and also about our institute. And then I will tell you a little bit about the world of nanoparticles, nanomedicines, drug delivery applications. And then I will give you a few examples how we can apply these materials for medical applications. So we focus mostly on engineering, and engineering with a clearly medical application and medical translation. And then I will have some conclusions. So to give you an overview, so this is Europe, and Netherlands is located over here. So it's a very small country, but with about 17 million people. And basically, if we look inside of Netherlands, you will see that Groningen is very far from Amsterdam. So Amsterdam is there, and Groningen is over here. So it's about 2 hours by train or by car. And our research department, or my research department, is, even though we do a lot of engineering, as you see here, we are actually part of the medical school. So it's a very unique kind of department, because we are able to be together with medical doctors, together with the clinicians. So we can do very translational work and work together with the clinicians on biomedical problems. So if you are interested to know more in detail about that, so we have different web pages and also a YouTube video where you can learn more about our research over there. But just to give you an idea, so the University of Groningen is divided in two. So we have the medical hospital, as you see here, and then we have the university. So the university building is that one. It was founded in 1641, so it's also very old. And basically we work together, and they are very close together as well. So here we have all the chemistry, physics, mathematics, and here we have all the health life science. So if we look just to the hospital, so you see how it is located. So we have everything. So from this side, we have all the clinical research. From that side, we have all the preclinical research. And I am the pleasure to be in the highest part of the building. So I am on the 13th floor, and that's where I am sitting actually every day. And I can see the whole city and the whole medical school. And because if you walk from one side to the other of the building, it's about 1 kilometer. So we say that it's the smartest kilometer because you have education, you have care, you have innovation, and you have research in a single building. So just to give you some numbers, so this is the university numbers. We are about 35,000 students, bachelor and master students. 25% of the students are international. You see that we have about 600 PhD, more than 600 PhD theses. And our hospital is actually very large. It's the second largest hospital in Netherlands after Amsterdam. We have 1,300 beds and more than 4,000 medical students. And we do a lot of translation research in the hospital. So we have clinicians working very close together with the researchers. And recently, we also created what we call the Health Technology Research and Innovation Cluster. So this cluster puts together different kind of research and education and innovation to impact. So it's everything about the impact. It's not just about the papers, but is how to research can impact the society. So we created this together with the engineering school, with all the applied schools, technological schools in our region, that we work together basically. And we are mainly focused on 4 areas. So you see here the areas that we are. Operating theater of the future, replacement and improvement on the human body, innovative technologies and local precision and point of care technology like biosensing and sensing technologies. Of course, these are all with the ideas of using biomaterials or robotics or sensing or medicine in order to tackle different medical problems in the northern part of the country. So when we look more closely to the department that I am leading, so this is our department. So these are all our professors that we have in the department, and basically our core research lines are these ones here. So we work a lot on advanced biomaterials and biofabrication, targeting therapies. So this is what I'm going to tell you today. We also work a lot on bioimaging and bioanalysis, particular on also on organ on a chip. So I will tell you a bit later. But also our interest is to understand how materials interact in the body with different cell applications. So I will give you some examples how actually we are doing that. So we always have a medical problem in mind, and we also work with the clinicians towards solving that medical problem. But we are also very committed, of course, to education and training. So we work a lot with engineering students, we work a lot with medical students, and they do research activities in our department in order to solve some of these medical problems. So because innovation is also one of our translation or impact is one key aspect in our department. Here I show you just one very successful spin-off company. So we actually launched 2 years ago an instrument that is quantum sensing, use quantum sensing technology in order to detect specific chemicals, in this case, reactive oxygen species in different cells or tissue. And this instrument is now actually available in the market, and it comes actually from our department. So it's a way out to go from the research lab actually to a precise equipment and precise instrument that is already in different countries in the world, including actually in FDA. So now let me tell you a little bit our research focus. So what actually we do in our lab our technologies and how do they help to improve the human health. So we have been for many years trying to understand how drugs affect the human body. Different drugs, this can be synthetic drugs, they can be natural-based drugs from different exotic fruits, from plants and so on, and to make them very precise. So traditional drugs, they are non-specific, and we want to make them more specific. And for this, we have been thinking of using, for example, nanoparticle technology. So the idea is that we have such kind of particles like this one here. We precisely target them to the tissue, to the cells, and then deliver the drugs inside of the tissues or inside of the cells. So having that in mind, we can use different kind of materials. So we have started in the past to work a lot with inorganic materials, so silica-based materials, magnetic materials, quantum dots as well. Organic materials, so lipid-based materials, liposomes, polymers, polymeric materials. And then we evolve and we start to use also what we call hybrid materials. So basically it's a mixture of both of these worlds, and there is a lot of potential on using this kind of combination. I will show you some examples. So basically we believe that small things are very powerful. And we say that small is actually more, and the size does matter, because once you go smaller, you can do a lot of things inside of the body as well. So our nanoparticles have been used in nanomedicine, and you see here some examples for drug and gene delivery, also for implants, tissue engineering, photothermal therapy, photodynamic therapy, but also for imaging and diagnostics. So we can also use those to imaging materials in different settings. It can be in a fish, it can be in the body, it can be in different kind of applications. And as I said, you can make hybrids, so you can use synthetic materials and you can use natural materials in order to combine this to make it more effective, more efficient. So we try to solve the problems in the body, but we know that in the body there are different barriers. You see here different kind of barriers where these materials have to cross or they have to move. And therefore, it's not so easy. It's not just you have the material and then it will do the job. No, you have really to make sure that you understand the biology very well, so that you can then engineer your materials to make them pass all these barriers. And basically, we have worked on all these kind of barriers in our research for the past more than 20 years, actually. So I just go to give you some examples. Of course, we are very interested to improve the human health. So typically synthetic drugs, so small molecules or large molecules or proteins, we like to put them inside of carriers like this one, for example, in order to, if more efficient, deliver them inside of the body. And you can think about oral delivery, but you can also think about the skin, or you can also think about injectable materials. I will give you several of these examples. So why this is very important for us? Why we want to do this? Well, because we can control many things of the properties of these materials. So we can control the targeting, the drug release, we can control the polydispersity of the materials. We can load not only one drug, but several drugs, for example, for combination therapy. And also we've been working more on scaling up technologies and also reproducibility, because what you want to do is that to make sure that your technology can be translated into companies, okay? So this is very, very important. So when I put it in a very large setting, these are the research works that my lab, my specific lab, is working on. So I'm showing you today just this part of the work, nanomedicines and drug delivery, and basic some of the cardiovascular disease. But we actually work on many different applications, as you can see here. So it goes all the way to tissue engineering, to regenerative medicine, theranostic properties as well. Okay, so we have then a bit of examples, and of course, we like to collaborate a lot also with the clinicians and with the industry. This is key to translate our technologies. And in addition to the collaborations that we have, for example, with Big Pharma, we have also established few startup companies. So I already helped 3 startup companies to be established for working in these technologies. So when I look to the research that we do, you see here some of the examples of our work. So we try to basically understand how the materials work and then try to apply them for different kind of biomedical applications. So you see here all the applications that we have done for the past years in different kind of fields: orthopedics, wound healing, cardiovascular disease, regenerative medicine, cancer, and now lately more on cardiac regeneration as well. So in other words, we try to understand how these materials are fabricated. We precisely fabricate these materials with different properties. So the physical chemical properties of these materials, this is what we try to engineering very precisely. And then these materials have to have some function. So they have to respond either to some external stimulus or some internal stimulus, such as, for example, pH, enzymatic reactions, or the heat or the light. So we want them to be very, very smart in the sense. So then we have work all the way from small drug molecules or small molecules down to now we are more and more interested in therapeutic nucleic acids. And this is the examples that we are going to use to you. So at least Professor Zhao knows this very well. So we have been working in the same lab, in Professor David Witt's lab in many years ago. And this is the one technology that I learned when I was in Harvard many years ago, also doing part of my postdoc. And this technology has been very useful because we have used this for creating all of our microparticles, nanoparticles, for everything that you are going to see basically in this presentation. It's called microfluidic technology, and many of the big pharma and other, even cosmetic industries using for producing emulsions, microemulsions, for example, for the cosmetic products. So we have used this. We have actually few patents also in China related with this, because I had very good students that brought this technology also to China, and then we patent some of this technology also here. But we have been very interested in new manufacturing methods. So 3D technology, we are doing quite a lot on 3D printing technology and also microneedle technology, special to delivering different kind of materials through the skin, drugs, food supplements, vitamins, you name it. So we can also do this kind of technology in our labs. So I didn't have time to tell you about everything, but I just give you some examples. For example, this one here is a work that we did. So we use microneedle technology to deliver oncolytic virus under the skin for immunomodulation. So we create a kind of a vaccine for cancer therapy by using this kind of microneedle technology, like I show you in the previous slide. So this is a way how you can transform a material for a skin delivery vaccine, for example. So currently we are working quite a lot on, sorry, go back. We are working quite a lot on 3D printing technologies, so where we prepare different kind of bioinks and inks in order to have different materials, special for wound healing applications, but also regenerative medicine like patches, human patches, and we create all these materials in the lab. And here I show you just some examples where we can 3D print in these materials very precisely with cells in between, so that you can actually then use them with patches on the skin or then inside of the tissue in the body, like for example in the heart tissue, for example. So this is just to give you some examples. So then when we take the whole picture of nano platforms, so you see here So very specifically, what kind of nanoparticles we have been used: lipid, polymeric, porous, hybrid, and EVs, extracellular vesicles as well. And we have used these platforms for different applications, so cancer, immunotherapy, cardio, diabetes as well, gene delivery. And again, we have different models. So we have developed different in vitro models in the lab, but also in vivo models. So we have different kind of technologies that we can use in order to translate our technologies, and as I said, to make some impact. So this is the main core of our research when it comes to nanotechnology. And as I said recently, we have been trying to use more natural materials. So we actually created what we call cell-based nanoparticles. So these are actually materials that we extract from the cells, the real cells, the human cells, in order to create particles that we can use them as vaccines, or we can use as drug carriers. So instead of using synthetic materials, we use actually our own body kind of material, biological material. And recently we have done all of that, so we can use different kind of what we call nanocarriers with proteins, different kind of proteins extracted from different cells. And we have mastered the technology, so we can very easily create this and understand how they are made, the characterization of all these materials, and understand if we use a different cell, what kind of particle we get. If we use a cancer cell, liver cell, immune system cell, what kind of nanocarriers we are able to create to deliver different kind of drugs. So this is what we call cell-based materials. And with that in mind, actually not very long time ago, we actually, before COVID, we created also a vaccine that was just based on this. So what you see here is actually a nanoparticle that the core of this nanoparticle is a virus. And the wrapping around is actually a cancer cell membrane from the animal. So we take a biopsy from the cancer, we created this kind of nanoparticle, and then we inject it back. And what you observe is that we could actually kill more efficiently the cancer. We also did with colleagues in Hangzhou, actually. So I had a very good collaboration there as well, where we use again the cell materials, and by doing so, we were able to demonstrate that these materials could also kill more effective the cancer. Pei Zhang was my PhD student and now also a postdoc. And basically, she has been working quite a lot on developing SARS-CoV anti-SARS-CoV nanoparticles. And basically, this is for pulmonary delivery, and we have been using quite a lot. Together, we also established a company, and this is now very well established technology in our lab, where we can use to prevent the virus to actually go to the lungs. It's very, very interesting, because in the morning we had a very good tour from Professor Wong in the lab about the food, the plants. So we also have used plants to extract particles. So we generate what we call a virus-like particle that is produced from plants. And basically, we can extract some of those plants. We infect the plants. They generate some plant virus. Those are very safe. We extract those virus. We convert them in nanoparticles, and these nanoparticles can be used to deliver drugs inside of the cell. And it's very, very interesting and very, very efficient as well. So we also did the one very recent for melanoma. And recently, we have been also working a lot with fish. But in this case, the fish was, it's a kind of our tool in order to understand how the materials work. So basically, we work a lot with zebrafish, embryonic zebrafish. And the zebrafish, we inject different particles in order to understand how our in terms of toxicology, nanotoxicology, the biodistribution of these particles inside of the body of the fish. And then we try to understand what kind of interactions and what kind of models and what doses we can give to the animals. Hu Ji-han was my PhD student. Now she's a professor in Beijing University of Technology. She has been working a lot on developing different kind of nanocarriers for cancer applications. I just give you some examples. I'm not going to discuss so that I can go. So we have been very much interested in producing reactive materials, such as, for example, these particles. They are porous structures, hydrogel-based structures that we can use. We inject under the skin in order to, they are the depots, and then with the light, we shine with the light, and by doing so, we can release the drugs because these materials are reactive with the light, and then we slowly release the drugs under the skin, and then they have some therapeutic effect. In this case, is for cancer. So she has been developing different kind of materials for different kind of applications. Then Jiangshan Li was also my PhD student. He's actually from Nanjing, but now he's a postdoc or a research assistant in Professor Chui's lab, actually. So he's back to China. But he was in my lab, he was very much interested on using materials, so a combination of hybrid materials, synthetic materials, with biological materials to create a responsive kind of therapy, vaccine therapy for immunotherapeutic applications. So he's really a brilliant student, and now he's back to China as well. And he has been working quite a lot for many years on also light-sensitive materials so that we can then apply, you see here, gold nanoparticle materials, ions delivery into the body in order to make them very reactive and in order to kill cancer cells in this case. Okay, so this was just a short introduction to get to the point of why then we got interested to the heart. So we started our research in the past on diabetes, trying to deliver drugs in order to decrease diabetes. Then we went to cancer. So many of the research that I showed to you was on cancer. And about 10 years ago, we started to work more on heart disease. Why? Because, well, you all know heart disease is the #1 in the world. It's still killing more people every day. It's because of the food, because of the exercise, because of many different genetic diseases as well, because of many impact that it has in the human body. And it's very tricky to treat because you only know the problem when the disease is already there. And that's what it makes it very, very tricky to solve. And also the heart tissue, it's very different than the other tissue. So the heart tissue is not able to self-regenerate. So it's another challenge that we have when we compare to other cells in the body, for example. So we have been trying from already for many years, try to understand what kind of materials you can use. So for example, we started to use patches, 3D printing patches in our lab. And these 3D printing patches, we added different materials. They are electroconductive materials that they contain nanoparticles or drugs that we can then put on top of the heart so that we can, after an injured heart, for example, myocardial infarction, or after a heart attack, the patients, they need to be helped, and we can put this patch in the heart tissue. They are electroconductive. They are materials that you can help the heart cells to regenerate. So we started to do that. We also started to do electrospinning of materials. So they are nanofibrils of gelatin that we prepare some patches as well, also with drugs that you can put on the heart tissue here very, very nicely after a surgery. And then the tissue is going to heal over the time. And on top of that, you can make them, as I said, electroconductive. So here you see some examples of, let me see if the video works. But basically what we demonstrate is that you can very nicely get these materials to be electroconductive. This is what you need also in the heart tissue, because the tissue has a special feature, and with this you can make it a conductivity, electroconductivity as well. So this is just some examples, and then we understand about the properties of these materials, the tissue, that they have to be biocompatible. They have to be, the tissue has to stand these materials that we put in the heart as well. So then lately, of course, what you saw means that the patient, we need a surgery in order to put those materials in the heart. So it's very difficult, it's very invasive. So we wanted to find a way how to make it less invasive. And less invasive, you can do it, for example, by working with nanomedicines or nanoparticles. So this is what we've been trying to generate recently. So we started with a very, well, I would say, very nice particle. So this particle here was a porous material particle. You see here porous particle. We had peptides attached, we had the drugs, and we also had these stars here. This is radio label, so we can follow the particle after we inject that it goes to the heart tissue, okay? So what happens then? So then you can precisely see, this is an animal, this is a rat, and you see here the heart tissue, and you see the accumulation of the particles into the heart. So we got 60% of the injected dose of the particles, they went to the heart tissue, which is really, really nice. And we saw some recovery of the animals over the time. So by having that in mind, then with the new technologies, such as the RNA-based lipids, so we started to develop new technologies so that we could even more precisely engineering these particles to the heart by using RNA, siRNA, mRNA, different kind of technologies so that we could improve the heart recovery. So these are some of the examples that I'm going to show you. So these are some of the students that I got over the time. So Zehol Yu was my PhD student, postdoc, now he's a professor at the University of Helsinki. We are still collaborating as we speak on the heart tissue. Han Gao was also my PhD student. Now she's a professor on Southeast University in Nanjing. And basically, they were our students working on this cardiovascular disease. So many of the studies that you are going to see now is actually based on their work. And because you talk about food, there is one component that actually we were using, or we are using, that is extract from food elements. So this is why I said there is a lot of things that we can do. So beta-glucans is actually a polysaccharide that you can find in different kind of food, for example, specific on mushrooms. So few years ago, this guy here, Zehol Yu, had an idea, or he read that beta-glucans had a specific healthy benefit. So he thought that why not convert beta-glucans into a nanoparticle and then put inside of this beta-glucan nanoparticle some drugs and then see what happens in the body. For our surprise, what we discovered is that beta-glucans, they actually attach to the monocyte, one of the monocyte receptors. And when they attach to the monocyte receptors, it can travel all the way to the inflammatory tissue. So this has been demonstrated with beta-glucan alone, but we had no idea if with nanoparticle it could work. So we did a lot of studies, and he actually demonstrated in this study to the heart tissue. So he demonstrated, he's a chemist, but he worked a lot with biology. This was a model that he actually developed. And what you see here is, this is the heart injury, and this is our cocktail with the beta-glucans. And you see that the heart actually has improved, has become more like healthy when we add beta-glucans. So we were very happy to see that, so happy that then Gao, so the next student, she started to use beta-glucans, but with gene delivery, so with RNA therapy. And what we observed we did different models. We did a liver model, we did a heart model, and we did also a pulmonary model. And what we saw is that these particles were able to actually navigate and attach in these tissues. So I will just show you an example of the heart. So these are beta-glucan nanoparticles. They are very small, 200 nanometers. They are very stable. And you see here that we can put RNA inside of these particles very, very efficiently. So when we look to the heart tissue, so this is a human animal model, but with a human model of myocardial infarction injury, heart injury, we injected 2 times, and at 7 days, we look into the heart tissue. And what we observe is that we have a decrease into this protein expression and also a decrease on the collagen. So it means that the fibrotic tissue was able to be decreased by using this kind of approach. So as you see, a food-derived material can also have a very influence in the health. We also demonstrated with other kind of combinations with inorganic materials and beta-glucan materials that they could also work. So this is MOFs, metallic organic framework. So they are very small porous materials. Well, you know, the Nobel Prize of Chemistry last year was on MOFs. So we were actually already doing some research. And when you combine it also with beta-glucan as well, you can have exactly the same effect. And I will just to show you some of the results. So here we use it for atherosclerosis. And what we demonstrated, so this is the atherosclerosis model. And this is after we did our nanoparticle with RNA therapy. And there is a huge recovery by using this kind of approach. Okay? So there is a lot of benefits how we can use it. So of course that was in the lab, and now when you want to scale up, it becomes very difficult. So what we did is that we created a very high-through microfluidic technology. So it's a very automatized microfluidic technology where we can create all these nanoparticles, and then we can also analyze the physical chemical properties of these particles in situ. So you don't have to do one experiment, wait several days, and then go back if you need. You can do everything in situ. So it becomes more efficient, and it becomes also more scalable. So we tried different materials, different polymers, as you see here, chitosan, polyethylene material, dextran. We also work a lot with dextran material, which is also food-related sugar, a lot. And then small drug molecules, peptides, and different lipids as well, synthetic lipids and also biological lipids. And then by doing this, we get a lot of, with our robot, microfluidic robot, you see, you get a lot of this blue and gray. So it means the blue are the good particles, the gray are not so good particles. Still, we have a lot of blue. So how we distinguish before we go to our animal studies? So for this, we use computational analysis. So we are also working a lot with AI technology or computation analysis so that we can screen down all this technology, all these nanoparticles to one good nanoparticle that we then test in vivo. So you don't have to do all the in vivo testing. You can then just have, with the help of the computational and AI technologies in order to create the best nanoparticle possible, okay? So then we did it in the heart again. So you see here some of our examples. So this is again with beta-glucan nanoparticle generated based on what I just showed you. And then you have we follow the animals for 28 days. And what I'm showing you here is the electrocardiograms. And also here, what I'm showing you here is the heart tissue. So we analyze the heart tissue. And when we go from here to here, so we see a very improvement on the tissue, on the wall of the heart. So if this becomes thicker, so that is very positive. So it means that the function of the heart is recovered and you can improve the functionality of the heart tissue. So this is very, very good. And with Z very recently, we had, we try a different approach. So we use nanoparticles derived from our blood. So we take our blood, there are the red blood cells, we extract some material from the red blood cells, and then we make a particle out of those. So it's called nanoerythrosomes. So they are basically blood nanoparticles based. And then we try to understand what happens when they are injected back to the body. And what we understood was that some of these particles, they accumulate a lot in the liver and in the spleen. And what Zehoa was very interested to understand is that if they go to the liver and spleen, can they still go back to the heart? And what we demonstrated in this study was actually very, very interesting, is that if we take a spleen from the animal, the particles still don't go to the heart. But if the spleen is present and the particles go to the heart, go to the spleen, they are still recycled and back to the heart tissue. So it means that there is a mechanism, and we try to understand. I just show you here again. There is a mechanism of the particles that even if they go to the spleen, they are actually attracted. So the inflammatory cells in the heart will take these particles, or they will kind of send the kind of response that they are taking then from the spleen to the heart tissue. And what you see here is a recovery again of the heart tissue, and the electrocardiograms after several days, they improve tremendously. So it means that what this demonstrates is that there is an improvement on the myocardial recovery outcome, even if some of the particles end up in the spleen. And one of my last studies that I wanted to show you is with machine learning. So you know that most likely machine learning now is very popular, and we try to apply also machine learning for designing the best possible materials for a medical application. So in the past, you have materials like I show you here. You have compounds, so we have several compounds, almost 900 compounds, and then you had to test one by one and see which one was the best. So this took many years, this took a lot of work, and now we can more efficient and more rapidly actually do this. So how? We use machine learning mechanisms to try to understand how these materials and the drugs or any compound, they interact, and which the best outcome you can get for the disease of interest. So in this paper here, we tested for cancer application, and we tested for heart. I will just show you about the heart tissue, but it works exactly the same way for the cancer. And then by doing so, we can design the material or the nanoparticle more efficient for heart tissue or pancreatic cancer, for example, in this case. So let me just show you what we did. So we start with a very simple particle. So this is a PLGA particle. We start with antibody drug. We generate these particles. We can see the particles, also imaging the particles. How do they look like? How much antibody we have inside of these particles? How small they are? How stable they are? And then we can inject them into the body and see, for example, where do they accumulate over the time. So you see, you can image them, so you can see very precisely in which part of the body they are actually. And as you see here, over the time, we see an increased accumulation into the heart tissue, for example, the inflammatory. So it means that the inflammatory tissue attracts these particles to the tissue, and there you can actually release the drugs of interest. So we actually checked with our drugs, and what we observed here over the time is that we could more efficiently improve the delivery of the drugs into the heart tissue and increase the response of the tissue. So the tissue behaved more as normal, basically. And this is what we want to understand. But as you know, some of the disease or some of the heart disease are generated by food. So by the food that we eat. And therefore, what we also try, or what Zehoa was trying to understand, is that if you eat or if there is a disease like diabetes, how that interferes in the heart disease overall. So we did a lot of omics in order to try to understand which kind of mechanisms the diabetes has in the heart, and then when we have our particles, which of this kind of where the particles actually influencing these mechanisms. So we study all the biological process to that point. So this is I don't have time to go into detail, but basically we develop a way how to understand, first of all, if the food generates diabetes, if there is a diabetes that generates heart attack, how then we can use our nanoparticles to tackle those events in order to biologically understand more from the protein, from the gene influence. And basically by doing so, you can actually make clusters of different mechanisms, of different effects. And again, you can then, your nanoparticles, for example, what you see here from 1 week to 4 weeks, we see a better improvement of the heart tissue over the time. So it helps you then to get the most efficient material in this case, for heart tissue. And I'm running out of time, so I have to go to the conclusions. But basically, what I tried to demonstrate here is that synthetic materials or food-related polysaccharide materials, or then even lipids or cell materials, can be used for, in this case, for heart regeneration, for myocardial infarction therapy applications. This is just one example, but we have several other examples for other diseases as well. So it is important to understand how simple materials that sometimes we just eat, like sugars and so on, they can have a very big impact in the disease or to treat disease if you engineering them properly in order to address a medical problem. So of course, this is all the research that I show you. It's only possible with a lot of cooperation. You see a lot of companies here from big pharma as well, but also the CSC, the Chinese Scholarship Science Council, has helped us quite a lot. We have had a lot of students from China coming to my lab, and our group is very multinational. So you see here a lot of students also from China, but from different kind of countries. We like to have students from all over the globe with different skills, with different knowledge. Because we do a very multidisciplinary work, and this is very relevant then to understand. So we have chemical engineers, biotechnologists, biochemists, chemists, physicists, mathematicians, in order to understand all the things that we do, in order to generate all the work that we do. So we are very, very happy to welcome everyone to this. And here we also have a special issue. If you like to contribute to this is a special issue that has been recently published. It's on biomaterials and tissue engineering, so it might be interesting for some of you. The impact factor of the journal is very good. It's 6.6, so it might be interesting also for some of you to submit your work here, or your colleagues you can address. And I'm also associate editor of NanoSelect. So I anything related with nanotechnology or materials in general, you are very welcome to submit as well. And I invite you also to submit. The impact factor at the moment, I think this year is about 5. So it's increasing very, very fast. So you are also, I'm very happy to host your papers there as well. And with that, I thank you all for your attention here, but also online. And I'm very happy to answer any questions related with work or related with science or with other kind of collaborations. Thank you so much for your attention.
So, hello everyone. So let's start from the online questions. So the first one is, what are the major challenges in translating RNA nanotherapeutics from preclinical models to clinical application for myocardial infarction or heart failure?
Yes, thank you. Thank you for the question. There are a lot of challenges, but one challenge is the stability of these molecules. So you have to make sure that RNA is stable when you formulate them into your nanotherapeutics. So this is the first biggest challenge. Then the second one is to have enough of these RNAs into your material, because sometimes, for example, siRNA, we are able to have a lot, but then large molecules like mRNA, for example, or circular RNA, then it becomes really challenging because you can have only 20% at best. And then, of course, then it means that you need to inject more of these materials that increase also toxicity. So stability and the amount of RNA into the nanotherapeutics is some of the biggest challenge, not only for myocardial infarction, but actually for other kind of applications as well.
Do you have one question actually following this question? So how do you target the cardiovascular tissues? So from the nanoparticles?
Yes, very good question. Thank you. So we modify the surface of the nanoparticles. So that is how we do it. We incorporate the RNA inside of these particles, and then we functionalize the nanoparticles with different kind of molecules. So we are basically mostly interested on small peptides. So we use some technology to discover the certain receptors into the heart tissue after myocardial infarction, and then we inverse engineering those in order to create small peptides, usually 9 amino acids, 10 amino acids, and then we functionalize the particles with those peptides that they are very more specific to the heart tissue.
The second question is, how do the nanoparticle delivery system overcome the biological barriers such as the vascular endothelium, myocardial cell membranes, and lysosomal degradation to achieve precise RNA delivery to the ischemic or failing heart?
Yes, this is a very good question. It's like the holy grail of nanomedicine, basically. So overcoming biological barriers is extremely difficult in the body. So you have different approaches. One, as I already mentioned, everything that I showed you were mostly injectables. So when you inject something to the body will see it as a foreign element. So when it sees it as a foreign element, goes to the liver and spleen. So if you engineer in a way that you have better targeting, more specific targeting, then you overcome this barrier. So you get more to the heart tissue. But as I show in this very last study, even if those particles go to the liver and spleen, in certain conditions, you can still make them go back to the inflammatory tissue. But the best approach is the more precise functionalization you have of these materials, the more you can get these materials into the tissue of interest.
Do they go to the all like kind of tissues with inflammation, or they only go to the heart?
So we always try that they go only to the inflammatory tissue. So they, of course, if your materials are not sufficient functionalized or sufficient effective, they can also go to the normal tissue. But this is not what we want. So what we want is to engineer in these materials very precise that actually they go only for the cells of interest, in this case for the inflammatory tissue.
And any question from the audience? So I do have some questions. So what motivates you to find a component like the beta glucan from the food? How do you find that component?
So I don't take credit for that one. That was actually Zehua Liu. So he was my PhD student, and he was reading about what kind of materials to use. And then he read somewhere that beta-glucane had some properties. So some biology, one in the body, the sugar itself bound to, in this case, to monocytes in the blood. So, but there was not. It was just the sugar, the molecule sugar, but there was not so much. There was no work at all on therapy, and not with the nanoparticles. So he started to think basically, okay, does it also work if I convert the beta-glucane into a nanoparticle? And that's where it got very interested, and that's how we actually then started to use more beta-glucane material in order to understand if it really can be used for attaching to those to that receptor, and also if it can be used for therapeutic application. So I would say that it was just a little bit of try and error. So we knew that this sugar had certain kind of properties, and then we tried to understand more if it could be used for therapeutic applications. But like everything in science, sometimes you discover things that you are less expecting. So we do it with beta-glucane. In the past, we did it a lot with the dextran. So we work also a lot with another sugar, dextran. Basically, acetylate dextran. We modify it, and we have also used it for different kind of biological applications. So sometimes these natural products became really very interesting. And as you know, natural products like sugars or from food, you can have a lot. So you can take it from different sources, and it can scale up much easier than the synthetic ones. And they are a bit less expensive.
Oh, yes. And also we have one question online. So how does the precision manifest in this new era? Does it refer to the targeted delivery, patient-specific RNA sequence, or personalized dosing regimen? Wow.
So how does precision manifest in this new era? Does it refer to target delivery? Yes. So yes. So it refers to two things. One, it refers precision to really targeting specifically to certain cells and certain tissue. So that's what it means. It means that this material can go to the heart tissue and only to the heart cells, no one else in the body. That's what it means. And then personalize, it's also personalized in the way that we can make it, as I mentioned, for example, for cancer therapeutic applications, we can bioengineering materials that we extract from the patient, and then we give back to that specific patient. So we can bioengineering these precise therapies with the natural source of the body of the patient itself. So this is what it makes it very personalized. So when we have a headache, we take a pill, but my body is different than your body is different than your body. So it doesn't take into consideration the person, the proteins, the genome, everything. By doing personalized, then we can target specifically that person. And that makes it a therapeutic much more efficient when we compare to the general therapies.
So I see from your slides, and you're using the mesacrylated hyaluronic acid. So do you use that to make the nanoparticles?
Yes. So we use hyaluronic acid for many different applications. So we use it for to prepare microneedles.
Yeah.
So 3D printing structures as well, but also we use it as a functionalization property as well. So on Tuesday, I'm going to show some results also with hyaluronic acid.
Yeah.
So for targeting.
but for the hyaluronic acid, because they're hydrogel, right? So once they're injected, they might actually absorb water and expand.
Yes.
So how can we control
so
that?
we have studied for many years a way how to avoid actually that. So if you combine with different kind of cross-linking properties and so on, so you minimize the swelling. So you can take these hydrogels into a syringe, you can inject, and you actually don't have so much swelling when it's on the tissue. So you can, by using different kind of chemical approach, you can prevent the swelling properties as well. So I will show you an example or a few examples on Tuesday where we can precisely control that, because of course you don't, well, there are two ways. One, you really want to swell, and the other one you don't, but you want at the same time that there is a degradation over the time. So you can also control that by using different kind of chemical approaches.
So by using different kind of cross-linking ligand.
Yes.
Okay.
Yes.
Any question? Yeah, great. You can use the microphone.
You have the microphone.
Yeah.
Thank you so much, Professor Santos. It's a great honor to meet you and also listen from your interesting talks. You have mentioned you have done some work related to beta-glucan, the food-derived materials related formulation development, and that's for the precise medicines and other for the targeting drug delivery. So if I understand correctly, it's mostly go to the systemic circulation by M cell-mediated uptake and actin-1 receptor, that kind of thing. So it also have a chance to do immunogenic reaction, and how you mitigate those kind of aspects when, and also if not only the M cells, but also enterocytes may have some influence on the uptake. So how do you mitigate those immunogenic implications may arrive? Yeah, thank you so much.
Yeah, thank you. Thank you very much. We just realized that we met in a conference in US few years ago, actually. We have a picture together at that time as well. So thank you for coming here, and thank you for the question. So there are two ways how you can do. One thing that we try to do is to use the materials and the immune response in our favor, because sometimes you can translate that immune responses in a very positive way. So we always study how these materials affect the immune response or the immune stimulation. So in many cases, like in cancer or cardiovascular disease, you might want one or the other. So by engineering these materials in a way, you can, for example, in cancer, you want an immunostimulation. So actually, it is good that if materials do an immunostimulation in the right place. So in the other case, in the heart, you want the other way around, because you want to suppress a little bit the fibrotic tissue and so on. So there is not a protocol how to do that. You really have to understand how the materials affect the immune responses and then use it in your advantage. So in all the studies that I showed you, we always try to understand how these materials affect the immune system. So if they regenerate, if they go more on the pro-inflammatory or anti-inflammatory processes, this is where we try to use it in our advantage. So if the immune system does not work in your favor, then you have to do some engineering. You need to change something in your materials. So the first rule is that you try to understand the effect of your materials in the immune system. So most of these materials that we use here, they have a positive response. And when we do to cancer, we want to do it the other way around. We want really to stimulate. So this actually is the benefit of nanotechnology, because we can use it in order to help the immune system to actually either to help to kill cancer cells or then to minimize the immune responses. So I say always that the immune system is extremely important, and by using the correct bioengineering formulations, you use it in your favor. So we don't fight against it. So we try actually to use it in our advantage either way. No, thank you.
Go ahead.
Professor Santos, I'm not a medical person at all. Do I understand you correctly? During your talk, nanotherapy has been used to treat cancer for the liver and the spleen, and perhaps the pancreas as well. Is it being done now through nanotherapy?
Correct. So nanotherapy has been used already for many years for different kind of cancer therapies. So the first product in the market was already in 95. So this was the generation that I showed you here are already the 5th generation of that first generation that was already given to humans in 95. And basically many of or most of the COVID vaccines, they were based on nanotherapy. So the COVID vaccination, they are all based on everything that you saw here.
Okay, my next question is, I was just talking to the person that's next to me, and he's from CUHK.
Yes.
And to my understanding, there has been free machinery donated by Li Ka-shing to deal with cancer treatment for the liver. And one of them is in CUHK. And one is in sanatorium, and another one is in Gleneagle Hospital. So they are all using nanotherapy? Because I'm not a medical person.
I don't know if they are using nanotherapy in those? Sorry?
They have different, yeah. Particular that machines, I'm not very familiar with working with that, but yes, CUHK has a tremendous footprints on nano-based therapeutics and nanoformulations for this kind of things. And also our Vice Chancellor is very famous for non-invasive cancer diagnostics, Professor Dennis Lowe. So yeah, we do have a lot of work on that, but for that particular machine, I'm not very familiar with.
So in our case, we don't use those kind of machineries. We use this kind of, they are nanoparticle based or medical, you can call it a medical device, for example. But basically, they are all injectable materials. So you can inject them all, into the bloodstream, basically. So they are very tiny particles that they are all the big pharma, for example. Big pharma is developing this kind of applications, for example, for cancer. Yes. So a lot of research is going on that, and a lot of this kind of research is already in the clinical practice.
What's the difference between nanotherapy and treatment through ultrasound?
So well, first of all, the ultrasound and the nanomaterials, they can be used also together. So one is not independent than the other. So they can be used together. But in the ultrasound, you actually can also work with bigger particles. So you work also with microparticles. But basically they are related, but ultrasound helps is a way out to locally make sure that the drugs also are released from these materials. So if we inject, as I mentioned, we base a lot of our therapy in the chemistry and in the biology. And with the ultrasound, you help them with external stimulus in order to deliver that. For example, ultrasound, these materials are very difficult to cross the brain barrier, to go to brain cancer, very difficult. So one way how you could increase the amount of these therapies to the brain is by using ultrasound. So by using locally ultrasound, then you can increase these materials to go more to the brain, and then of course it becomes more efficient.
So you are saying medication is something physical?
It is very physical. It is very physical, yes. Yes.
But
So
nano is so tiny,
it is so tiny, yes.
extremely tiny.
That's why it's so beautiful. Because even though it's so tiny, we can do a lot of things with it. Because those tiny, even though they are very, very tiny, we have learned how to modify those by doing different chemistry, by doing different engineering in those very tiny things. And this is why it makes it so exciting, because even though it's so tiny, you can still do a lot of things. Just to give you an idea, a small nanoparticle, the surface area is huge. So it's several football fields. So you can do a lot of things, because once you go small, the surface area increases a lot. So then you can see, imagine in a football field, how many things you can do on top of it. So this is, even though it's very tiny, you can do a lot of things on that surface. You can put drugs, you can put food elements, you can put a lot of different things there. And then, well, you can engineering for the good of the health, for example.
What is smaller, nano or neutrino?
Neutrino is smaller. Nano is a bit larger than that.
Mm-hmm.
Yes.
There is an observatory in Jiangmen called JUNO, that stands for Jiangmen Underground Neutrino Observatory. Have you heard of that?
Yes.
Yes.
Yes, but those are very small elements. No, the nanoparticles the nano are a bit larger than that.
I don't understand why they put it there, because I went outside to visit it, and it only started operating a little bit over a year ago. And it's perhaps the most advanced one. I don't know why. It's
most likely, yes. We have also some in Europe as well, but you need a special for those. But these ones you cannot create it in such kind of places. So you really need specialized labs in order to create this one. Also because there is when you apply it, you can apply it for food, you can apply for animal, or you can apply for human, and then you need special places how to create those.
Thank you very much.
You are very welcome. Thank you.
All right. Thank you. My question is not very technical. In the initial part of the presentation, you gave us a very nice layout of your university, wherein you have different departments and you guys work harmoniously to produce a nice melody, so to speak. I work here at the Industrial Center. My name is Virag. One of the direction that I work on is micro 3D printing. So I do fabricate micro needles as well as microfluidics. At PolyU Industrial Center's perspective or direction is engineering based. So we don't really do independent research. We cater to different academic departments. Professor Zhao is also one of our collaborators at IC, a Science and Nature team. So now is that I have these microfluidics and microneedles, and based on the research that you just showed, the technical part, which is the fabrication part, is something we take care of. So on the high level, so in the initial part, you said that we do have nice collaboration with different institutions in your university. From the managerial perspective, how do we take it forward? Like an engineer, how does he take this thing forward to establish sort of academic collaborations to promote research using this sophisticated microfluidic device, or for that matter, microneedles that I have prepared?
You mean between your and us, or
No,
between
this is the industrial center
general?
in the PolyU campus itself? How do we promote the more usage? It's just from a managerial perspective.
Well, that's a very difficult question. I can tell you how we do it in our case, but of course inside of PolyU, I don't know very well how the system works. But usually in our case, so all this first of all, if you have any unique technique, you try to find a way of how useful it can be for different kind of research work. So if microneedles, so I see that I will already give you some good ideas because we discussed these past days. But for example, skincare, right? So you can use microneedles for skincare. Therefore, you can collaborate, or you can find those researchers that are working on skincare, but they're not using microneedles. And microneedles can be a way how you can also use it for skincare applications. Another way, I will give you another example, microfluidics technology. So in many of the creams, the facial creams that you put, like David Witt's actually, so Chanel and so on, they have microemulsions. Those microemulsions are prepared by microfluidics technology, and they are in the Chanel creams as well. So then you have to discuss with those people working on that kind of cosmetics, and therefore you can tell them, well, by the way, we have this kind of technology. You can understand better how to prepare this kind of micro or nano emulsions so that you can then use it in your creams. So that is how your tool becomes relevant to different kind of applications. So you need to find the right people that can also benefit from the technology that you can use. And that's how we do it as well. So we always try. So we were working on nanotechnology, and then there was an interest from a clinical point of view, in this case, cardiologists. So we try to discuss with them what are the problems, how we can help you. And that's how we became then doing these nanoformulations as well. Cancer the same way. So microneedles technology or microfluidics is exactly the same way. So we are also working a lot with those technologies for different kind of applications as well. So you have to find the right customers, so the right people that have they might not know how to do your technology, but they can benefit a lot from your technology, but you have to present them. So I gave you two simple examples that if you discuss with those people, I'm sure you will find a way how to collaborate with them, for example. But that might not be inside of you, PolyU. It can be also in other departments. So, for example, in the medical department, or in the veterinary department. So you probably know that, for example, microneedles are also a way out that you can deliver medicines to animals, because animals, they don't take pills. It's very difficult to take pills. So if you put these patches on the animals, and one of our first companies were actually based on this, is microneedle technology to deliver medicine to vet care. So you can always find a way how to collaborate if you find the right people that have a need of your tools that you do in the lab. So this is also how we try always to go around and to try to collaborate synergetically. So we have the tool, they have a need, and then we try to combine, and then we achieve some final product. I hope I answered your question. But internally, I have no idea how it works in your case, but at least that's what I usually even tell to the students. So I always encourage all my students to, if they are chemists, don't discuss with chemists. Go and discuss with pharmacists, with food chemistry and so on, because that's how you, by discussing with very different people from you understand, oh, you have something that I can help you. And that's how the best collaborations then can appear. Because if you are doing microneedles and someone is doing microneedles, there is no benefit there. So you have to find the complementary from the other different people.
We have one question from the online. So it's regarding your sugar.
Yes.
So that one is like, you mentioned the food-derived nanoparticles, and that can enter the body. Do you have evidence that they can offer the GPS capacity?
Interesting question. Thank you. So the materials themselves, we already tested. And the simple materials, no, there is no capacity that they actually offer by themselves the GPS capacity. So the GPS capacity we can only achieve if we can more efficiently achieve if we actually modify, functionalize the surface of those materials. So by the materials themselves, we have not found yet one, but we don't know. We have not tried all of them, but at least those that we have tried, we have not found one that by themselves have enough GPS capacity that can be used. So we always do a further modification, a further functionalization on top of those materials in order to have the most efficient GPS capacity. So we would love to find a material, we would love to find something, a protein, a peptide, a polymer, a sugar, anything that would give by itself that that GPS capacity, but so far we have not found. Sometimes by combining some of the materials we can get a better effect, but it's still not enough. So we always need to go even further, and further means that you need always a step to functionalization. Why this is very relevant? Because if you would find such thing, you don't need to modify. That means you drop the costs and you can scale up way much better. So this is where we struggle a lot, because once you try to modify, you increase 10 times the cost, and also you need the manufacturing becomes more expensive, and also the scaling up becomes way more expensive. So finding those kind of materials would be great. I don't know, maybe in food there is, maybe in fish there are some materials to be used, but yeah, so far all the screening that we have done, we didn't find one that was enough effective for that.
So due to the time limit, last question. Go ahead.
My research was focused on the nanocarriers on thrombosis disease. So it is connected with Dr. Santos' research, and I've used hybrid materials and lipid and polypeptides, but I never used cell materials. So I want to ask, is the cell materials make the carriers less exclusive, so it won't be easily cleared by the immune system or the spleen? So the therapy would be more efficient, or there is any other advantages of the cell materials?
Thank you very much. That's a very good question. So there are different kind of qualities that why we use cell-based materials. So the first one is that we make it very personalized. So we use materials from a person, and then we give back the same material to that person. So then you have less clearance, less potential immunogenic responses, and yes, more effective distribution and delivery of the drugs inside of the body. So, however, if you want to do it to produce massive of that, so scaling, that's not an option. Okay, so it works very well if you want to do it on a personalized effect, but if you want to do it for a large population, that's going to be very difficult and very costly. So, but one of the properties is that you make it very specific for the patient, and you make it more compatible to the patient. So there is way less toxicity, way less immunogenic reactions, and that is a great benefit when we are doing this for medical applications. So this is the most important use of cell-based therapies.
Okay, thanks very much.
Thank you.
Kevin, you have one question. Go ahead.
Oh, sorry, I just have a quick one. So for the nano emulsions that you make, how stable are they? Do you need addition of like emulsifier or other chemicals to preserve them?
No. So the very good question. Thank you. So you don't need any additional emulsifiers. So we basically already stabilize them with the surfactants in the very beginning. So we use some surfactant to actually stabilize them all the way. So once you produce them, they are already stable. They can stay for a long time. So after that, there is no need of post stabilization on that. So for example, a very easy way is that we use PVA polymer, and this PVA makes it actually very stable. And therefore, once you have it in a suspension, it can stay for a long time. So it's actually very stable. Thank you.
Yeah, thank you very much.
No, thank you very much.
Also, let's end the section now. Thank you very much. Thank you very much for the speaker, and also thank you very much for coming.
Thank you. Thank you for coming. Thanks.
Okay.
So we also need to, sorry, I'll stay here. So we also need to present the souvenir.
Yes. Okay. Thank you so much for Professor Santos' interesting and inspiring talk. I think I really learned a lot. And in fact, I also have many ideas already in my mind for collaboration from a food scientist point of view. So, I'm working on nanoparticle, but it's edible one. So, right. Okay. So without further ado, we have some souvenir for you.
Okay, perfect. This. Do the solvent vertically. This. Perfect. 1, 2. Thank you so much. Thank you.
Okay. I think this is the end of this seminar. Thank you for coming. Okay, see you maybe next one. Okay, for the pair seminar series. Okay, thank you very much.
It makes sense.